Hey guys, so today we'll be talking about the immunology of diabetes mellitus type 1. So we'll be talking about all these things. Start off with central tolerance and then peripheral tolerance, and the stages of type 1 diabetes, how it occurs, T regulatory cell formation, and then different factors that can increase your chances of getting type 1 diabetes. And then we'll end off with some new treatment that is underway. Okay, so we have the bone, and in the middle of the bone we have the bone marrow, and the bone marrow releases a lot of different types of blood cells, including red blood cells and monocytes and macrophages and T cell precursors. So these T cell precursors are trying to go to the thymus, and the thymus releases thymic factors which attract the T cell precursors towards the thymus. And the thymus is kind of like a school where the T cell precursors grow and mature into adult T cells. I don't think that's actually a thing. You don't actually call it adult T cells, but like mature T cells. Okay. So let's say we're in the thymus right now. And so this baby T cell is in the thymus, specifically the cortex, which is the outside part. And it's given a backpack with lots of different things, including a TCR receptor and two CDs. So CD4 and CD8. Right now it's double positive because it has both. Okay? And so it has to go through the first exam in the cortex. And that exam is called positive selection. And this positive selection occurs in the cortex. So it's given two MHCs, and it has to try to open those doors using the locks that they're given. And so MHC 1 would require CD8 and the TCR, the T cell receptor, and MHC2 would require the CD4 and T cell receptor. So this T cell precursor, this baby T cell, is supposed to attach or be able to open one of these. Okay, and whichever it manages to open, it keeps that key. Okay, so if it opens it, they say, go on to the next stage, you passed the exam. Okay, and if it does not, then that baby T cell dies, or apoptosis. Okay, so if we see this illustration, we can see that the T cell receptor binds to the MHC class 1, and therefore in this case CD8 would predominantly be expressed and CD4 not so much, and so it would become a CD8 positive T cell. And if it fails to bind, it dies. Okay, so now we're in the medulla, which is the middle part of the thymus. And so this new exam in the medulla is called negative selection. So every baby T cell that gets to do this exam has already passed the first one, which is positive selection. So it's passed the first exam, and now it's on to the next exam. And so in this next exam, it's given lots of different doors and has to try to open one of them. But the catch is, if you open one of them, then you're in trouble. And these doors represent all the self-proteins. So all the different proteins that we don't want our T cells to recognize and destroy, because we don't want to destroy ourselves, right? And so if it does not open any of these doors, then it's fine. It's safe. But if it does, then you might be in trouble. And this is illustrated in this more scientific way of showing. If it fails to bind to a self-protein or a self-antigen, then this T cell is safe and it's not going to attack any of your own proteins. But if it does bind strongly and there's a strong connection, it's going to release Phas, which I'll talk about later, but it will lead to apoptosis. Okay? And so there's actually different options for this T cell now. So let's say this poor baby T cell managed to have a strong attraction with one of the self antigens, or one of the doors. And so this is a medullary thymic epithelial cell, and this is a self antigen or a self protein, and there's a strong connection. Okay, so medullary thymic epithelial cell. Now, the medullary thymic epithelial cell has a lifespan of 8 to 10 days. And therefore, because there's a lot of cells dying at a fast rate, there would be fibroblasts due to wound healing. And fibroblasts release TGF-beta. And so TGF-beta would target these T cells, these autoreactive T cells, and convert them to natural T regulatory cell. So basically, because this T cell is a potential danger to our body, there's different mechanisms to convert it into the good guy. It's kind of like how in Nazi Germany, Hitler found criminals and turned them into the SS, which was like his personal police. So it's like if there's someone who you know is going to be a bad guy no matter what you do, you could find a way to take advantage of them by turning them into a police instead. I don't know if that's a good idea, but in this case it is. So that's called a natural T regulatory cell. But if there's no TGF-beta, then this cell will be killed off. So that apoptosis. Or it will escape. So either they'll turn into a natural police, or they'll be killed off, or they'll be really sneaky and escape. And so we have the sneaky T cell that just escaped. Now, what are we going to do? Either outside of, so that all that I described before was central tolerance. Central, which is like the center, which is the thymus. But now that it's outside, there's still some hope of controlling this thing. So either it encounters a lot of TGF-beta somewhere outside the thymus, and then it converts into an induced T regulatory cell. And so this would occur in the periphery, not inside the thymus. But in the end, they would turn into a police induced T regulatory cell, which is good, which we're happy. Or this sneaky T cell will try to attach to, or will try to create a connection with an antigen presenting cell and the self antigen. However, because of the lack of co-stimulation, because there's no infection, so there's no co-stimulation and there's no cytokine release, and so the antigen presenting cell will be like, wait, are you sure? Because the way T cells are activated, we know that T cells, their action is very, very strong. And so before it takes any action, the antigen presenting cell has to ask the T cell 3 times, are you sure? Which is this one. And then a second, are you sure? And then a third, are you sure? If it's yes, yes, yes for all 3, that means, okay, actually, there's a problem. We need to do something about it. Activate T cells. But because there's no other infection, there's no other problem going on, there's not going to be any co-stimulation, and there's not going to be any cytokine release. And so this T cell will keep going to the antigen presenting cell like, oh my God, there's a problem. Activate me. And the antigen presenting cell double checks, and it's like, no, actually, you're lying. So it'll be like, how many times are you going to lie to me, you know? So the antigen presenting cell eventually tells it like, no, khalas, I'm not going to listen to you. No matter how many times you come and try to attach and try to activate yourself, it's not going to happen. So no matter how much it tries, eventually you'll develop something called anergy. So no matter how much this T cell tries to, and you get activated, it's not going to happen because it lied too many times. So that's anergy, which is also good. And this is called peripheral tolerance. So these are the two mechanisms of peripheral tolerance. Now, there are, and there's a rare chance that the T cell will be able to escape these two as well. And that's when autoimmune diseases come in. Okay, so there's different factors that lead to autoimmunity. Okay, so first of all, there's genetics. And so people who have HLA-DR3 or HLA-DR4 are more likely to have type 1 diabetes because these HLAs are more attracted to the insulin protein. Like they're more attracted. It's like if a mom had lots of kids, but she has that one favorite kid, she's going to take that one favorite kid everywhere, you know? So it's like this HLA really likes insulin, so it's going to attach it a lot and it's going to present it a lot. It's going to present it much more. And so it's more likely for this T cell to attach and get activated. Okay? Yeah, so increased affinity to binding to insulin. And so that increases the T cell's chances of finding it. But oh yeah, there's no co-stimulation. And so a trigger is needed. It's like, okay, where's your other are you sure? Are you sure? Are you actually sure? Do you actually want to get activated? You know? So there needs to be a trigger. And what is that trigger? How do you get that co-stimulation? For example, if the person is sick, that means there's another pathogen. And so the antigen presenting cell would have co-stimulation and it would allow like, what's it called? It would allow the 3 are you sure's, you know? Or someone fell and they got hurt. Okay? And that would lead to apoptosis. So increased apoptosis means there's increased inflammation, and so a lot of immune cells are out and about attacking all the pathogens. Okay? And so the antigen presenting cell would connect with this T cell, and there would be the 3 co-stimulations, and so it would get activated. Woohoo! Activated. Okay, so this is now the stages of T cell of type 1 diabetes mellitus development. Okay, I love you too. Now stop texting me. Okay. So stages of type 1 diabetes mellitus development, okay? So initially in life, your pancreas is fine, your beta cells are fine, everything's fine, but you still have your HLA-DR3 and 4, okay? But there'll be a precipitating event, like you might get sick or you'll fall, okay? Oops. Okay, and so that will cause the T cell to get activated. And once it gets activated, it'll start eating away at the pancreas. It'll start eating away at the beta cells because the beta cells show insulin on their MHCs. And so it's more likely the T cell is more likely to recognize it and so destroy that beta cell. And so slowly, eventually, more and more of the pancreas will get eaten away by the T cell. And so insulin levels will start going down, 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 until you get overt diabetes, where you don't have enough beta cells to release insulin. But C-peptide is still present because C-peptide is a part of the insulin protein that is released in the blood. And so if you check a person's blood and they have high insulin and they also have high C-peptide, then you know that insulin is intrinsic, whereas like if you give extrinsic insulin and you check C-peptide would be 0. So eventually you reach a point where no extrinsic insulin is being made, and so this person has to take insulin. And so, but they would have no C-peptide because it's extrinsic. And that usually happens around 12 to 14 years. Okay, now, can we solve this problem? There's this thing called personalized medicine, which is when you create the solution based on the individual. So based on the individual's genes and things like that. And there's this cool guy called Jeff Bluestone who thought of a way to help children with type 1 diabetes mellitus. So here is how he did it. He took children who did not have type 1 diabetes yet, but they had like a family history or their genetics were seen and they had HLA-DR3 and 4. Yeah, so anyone with HLA-DR3 and 4, they took the individual kid, drew out their blood, and then looked for the specific T cell that was the culprit for like insulin for type 1 diabetes. They took the culprit out. They put him in a petri dish, put TGF beta on this guy, and turned him into a police. And then they gave this police a lot of proliferative cytokines and created a lot of polices. And then they put all of these all of these T regulatory cells back into the blood. And so this kid would be fine in the long run, and they would not. So it's kind of like peripheral tolerance, except you force it, like you make sure that it happens. However, this method is very expensive. So, gotta keep that in mind. And that's it. Thank you to Dr. Farhan for teaching this to me. He's an amazing doctor and teacher, one of the best. And if you're a Muslim, you can read this, which will help you remember everything that you learned. Thank you, and I hope you learned something from me.